In Part 2 of cellular respiration, we will take a closer look at the 4 processes involved in aerobic cellular respiration. These 4 processes include glycolysis, the transition reaction, the citric acid cycle, also known as the Krebs cycle, and oxidative phosphorylation, which includes the electron transport system coupled to chemiosmosis. In glycolysis, you can think of it as literally splitting glucose into 2 molecules of pyruvate. So literally, it's the splitting or lysis of sugar. Glycolysis occurs in the cytoplasm and has 2 major phases, the energy investment phase, or energy requiring phase, and the energy payoff phase. Please also note that in addition to occurring in the cytoplasm, glycolysis is an anaerobic process, meaning it requires no oxygen. In this diagram, you can see the 2 ATPs that are necessary to get the reaction going. This occurs in the energy investment phase. The energy payoff phase yields 4 molecules of ATP. In addition, reduces 2 NADs into 2 NADH molecules. Overall, glucose has been broken down into 2 pyruvate molecules plus 2 molecules of water. So we have a net gain of 2 pyruvate, 2 waters, 2 ATPs, and 2 NADH molecules. Remember that pyruvate is a 3-carbon compound. It might be helpful for you to trace what happens to the carbons from glucose, a 6-carbon compound, breaking down into pyruvate, a 3-carbon compound, and follow what happens to the pyruvate as the process continues. Before the citric acid cycle can begin, the pyruvate must be converted into a form that can enter into the Krebs cycle. This form is referred to as acetyl-CoA, and this will link glycolysis to the Krebs cycle. Sometimes this reaction is referred to as the transition reaction. In this diagram, we can see that the transition reaction will include pyruvate being converted to acetyl-CoA that will then be able to enter into the Krebs cycle. The pyruvate located in the cytosol was produced by glycolysis and needs to be transported into the mitochondrion. The transport proteins located on the outer membrane of the mitochondrion sometimes require ATP to shuttle them into the mitochondria. In this process, the pyruvate loses a carbon in the form of carbon dioxide. During this process, electrons are stripped from the pyruvate and are given to the NAD, reducing it to NADH. Also during this reaction, you can see in step 3, coenzyme A is added, forming acetyl-CoA that now can enter into the Krebs cycle. Note also that the acetyl-CoA is in the internal portion of the mitochondria, the matrix. The citric acid cycle occurs within this mitochondrial matrix. The cycle will oxidize the organic fuel derived from pyruvate, which is the acetyl-CoA, and during one turn will make 1 ATP, 3 NADH molecules, and 1 FADH2 molecules per turn. In this diagram, you can see the pyruvate located at the top of your screen that's been produced by glycolysis. Note the 3 gray balls representing the 3 carbons in pyruvate. Quickly, during the transition reaction, a carbon is removed in the form of carbon dioxide that allows the formation or reduction of NAD into NADH. Additionally, coenzyme A is added, forming acetyl-CoA. Note that the acetyl-CoA now contains only 2 carbons. To complete this breakdown of this organic fuel, it will now enter into the Krebs cycle, losing its coenzyme A, forming citrate or citric acid as the first step. As it completely is oxidized, 2 molecules of carbon dioxide are formed, 3 NADH molecules, 1 ATP, and 1 FADH2 are also formed. So the citric acid cycle or Krebs cycle has 8 steps. It's not necessary for you to memorize the products of each of these steps or the intermediate steps, but please note that each reaction in the citric acid cycle is catalyzed by a specific and unique enzyme. The acetyl group of acetyl-CoA joins in the cycle by combining with the last part of the cycle, the molecule referred to as oxaloacetate. This forms citrate. hence why we call the citric acid cycle the citric acid cycle, because the first product formed is citrate or citric acid. The next 7 steps of the cycle will decompose the citrate back into oxaloacetate, making this entire process a cycle. The NADH and FADH2 produced by the Krebs cycle will then relay their electrons that have been extracted from the food to the electron transport chain. Returning back to this diagram, we can see that we will not just have one pyruvate, but 2 pyruvates per glucose enter into the citric acid or Krebs cycle. So referring to the blue box, after 2 turns, which would account for 1 for each pyruvate formed from 1 glucose, it would yield 4 carbon dioxides, 2 FADH2s, please correct my diagram, 2 ATP, and 6 NADH. If you've been keeping track of the product so far, the 4 carbon dioxides plus the 2 from the transition reaction show that we've completely oxidized the glucose, forming 6 carbon dioxide. Think back to your overall equation of cellular respiration. The 2 FADHs will be used during the electron transport chain. 2 ATPs plus 2 from glycolysis gives us a net gain of 4 ATPs so far. If we're going to make 38, we know that most of our ATP will be made during oxidative phosphorylation, the next step. And a total of 10 NADHs have been formed, 6 from the Krebs cycle, 2 from glycolysis, and 2 from the transition reaction. So following glycolysis in the citric acid cycle, the NADH and FADH2 account for most of the energy extracted from food, or in this case, from the glucose. Remember that we have 10 NADH molecules and 2 FADH2 molecules. These 2 electron carriers will then donate their electrons to the electron transport chain, which will power the ATP synthesis during oxidative phosphorylation. The electron transport chain is located in the cristae of the mitochondria. The cristae is another name for the infolds of that inner membrane of the mitochondria. You may want to make sure that you understand what the structure of the mitochondria is. Most of the chain of the electron transport chain's components are proteins, which exist in multi-protein complexes, as we will see in the next slide. The carriers alternate reduced and oxidized states as they accept and donate electrons. These electrons being dropped off by the NADH and the FADH2 will fall as a free energy drop down the chain. Each of the proteins will be reduced and oxidized as they give off those electrons to the next protein. The electrons, remember, are finally passed to oxygen, acting as the final electron acceptor, and form water. The following is a diagram of the electron transport chain. You can see the purple proteins that vary in size from the large proton pumps to the smaller protein complexes. The smaller protein complexes are what will relay the electrons from the NADH and FADH2 down the chain. The large proteins that you see act as proton pumps, which will pump the hydrogen ions from the matrix into the inner membrane space. The electron transport chain in and of itself produces no ATP. The chain's function is to break the large free energy drop from food to oxygen into smaller steps that release energy into manageable amounts. In chemiosmosis, the final part of oxidative phosphorylation really couples the energy or potential energy produced by the electron transport system to the synthesis of ATP. So the electrons transfer in the electron transport chain causes the proteins, remember the large purple proteins in the previous diagram, to pump protons or hydrogen ions from the matrix to the intermembrane space. Really try to picture what this is looking like. The hydrogen ions or protons then move back across the membrane down their concentration gradient, passing through channels in another protein complex referred to as ATP synthase, sometimes just ATPase. The ATP synthase uses the potential energy of that proton motive force, that exergonic flow of hydrogen ions into the matrix to drive the phosphorylation of ATP. This is an example of chemiosmosis, how the cell can use energy in a gradient, this time a proton gradient, to drive cellular work. The cellular work here is the production of ATP. The ATP synthase molecule is shown here. It looks a little bit like a motor and a turbine combined, and that's pretty much how it works. The buildup of protons in the intermembrane space, remember that was caused by the electrons being dropped off at the electron transport chain by the NADH and FADH2. As they drop off the electrons and the electrons flow down the electron transport chain, hydrogen ions are pumped from the matrix into that intermembrane space. This creates a buildup of protons that then want to move back into the matrix. They want to move down their gradient. They can't do that because they have a positive charge, and they're going to be restricted by that phospholipid bilayer. They can, however, flow through certain channels like the ATP synthase. The ATP synthase will allow the hydrogen ions to move from the intermembrane space into the mitochondrial matrix. As they do so, it's sort of like water flowing across the top of a water wheel and causing it to spin. As the hydrogen ions move through the ATP synthase, the molecule will rotate, creating mechanical energy that allows a phosphate to be added to ADP, creating ATP. So the energy stored in that hydrogen ion gradient across the membrane coupled with the redox reactions of the electron transport chain allow ATP synthesis to occur. This is chemiosmosis. The hydrogen ion gradient is referred to as a proton motive force, emphasizing its capacity to do work. In this picture, we're kind of putting it all together. We have the electron transport chain with some of the proteins removed. You have the large proton pumps. You have the NADH and FADH dropping off their electrons. Note where NADH and FADH2 are dropping off their electrons, because NADH will generate 3 ATPs per molecule, where FADH2 will only generate 2 ATPs per molecule. You have the buildup of hydrogen ions in the inner mitochondrial space. You have the ATP synthase allowing the hydrogens to move back down their gradient, creating the energy necessary to convert ADP back into ATP. So during cellular respiration, most energy flows in this sequence, from glucose to NADH to the electron transport chain, creating a proton motive force that allows ATP to be regenerated. About 40% of the energy in a glucose molecule is transferred to ATP during cellular respiration, making a grand total of about 38 ATP. You may want to stop on this picture and make a tally of all the products that are being formed. Please note that how ATP is made during glycolysis and Krebs cycle is referred to as substrate level phosphorylation, and that's different than how the ATP in oxidative phosphorylation is produced. Also, because we make a total of 36 to 38 ATP, think about why that's so. Why it's not just 38, why sometimes is it 36 versus 38. So thank you for listening, and I hope that this has helped you understand cellular respiration a little bit better.